something that will be covered by his talk. So please, Thorsten.
Bueno, ¿me escuchan? Bueno, muchas gracias, Fernando, para esta invitación. Y para mí es un honor inmenso dar básicamente la primera charla científica de este congreso. Así, muchas gracias a mis amigos españoles. Bueno, so I welcome you all to my talk about spatial point pattern analysis in ecology. And a brief outline of my talk. I will give you some basics about spatial patterns and processes in ecology, some basic things about point pattern analysis, and then some examples of my current research about my bigger EEF project. I will try to explain to you a bit how one can quantify complex univariate structures in point patterns, something about co-distribution patterns in species communities, some ideas how one can quantify spatial structures in diversity, and how to integrate dynamic simulation models in spatial analysis. Yeah, space and ecology. Many years ago, there was no space in ecology. So most equations could not handle space. Experiments try to avoid the influence of space. But the whole thing changed about in the 80s. And then it became recognized that spatial structures and processes are quite important in ecology. So just a few of the main themes we are facing at the moment, and they all are closely related to spatial things. So the big question, why do organisms appear in one location and not in another? Or will organisms change their distribution in response to climate change? And how does this depend on this person? Another critical question is, what's the impact of habitat fragmentation on population viability? And then the question which I will talk about today, which processes allow persistence of hundreds of species in tropical forests? So you see, the current ecology is full of questions and things that are related to space. So it may be a quite important thing to quantify spatial structures. Yes, especially in plant communities, it's well known that most species are randomly distributed. They are usually clustered at one or several spatial scales, and also many persistence mechanisms have a very strong spatial components. For example, competition is an inherently spatial thing, or the Janssen-Cohen hypothesis, or many other things. They are closely related to spatial processes. So therefore, if these spatial processes are important, they also should leave some type of signatures that emerge in spatial patterns. And if this is the case, one can have some hope that one would be able to reveal the underlying processes using spatial analysis. Yeah, in a way, this is a justification for my work, for the work that tries to quantify spatial patterns. Yeah, and then there are a number of tasks. The first, most basic task is to describe the spatial patterns as close as possible. And this is not always a trivial task, because often the structures are really complex. And then, based on the description of the spatial patterns, one needs to derive hypotheses on the underlying processes. And then in the final step, of course, one can test hypotheses on these processes. Yeah, and one important technique which became quite strong over the last years, but which is around for many years, at least 8 of the 70s, this is point pattern analysis. Yeah, what is a point pattern? It's a relatively simple thing. Point patterns are composed by the coordinates of ecological objects in a certain observation window. Then perhaps you may have some information attached, like if you have trees in a forest, you may have the size of the trees, you may have the condition of the tree, like surviving or that tree, or you may have the information of species. And additionally, you may have some environmental covariates. that may influence the distribution of the points in space. Yeah, here's one very simple example. This is a univariate pattern. This is only one point, one type of points. And now the same pattern with the mark size. And here you see that there are some additional interesting spatial structures emerging when one looks at the size of the trees in this example. And then one can have two types of points. In this case, it's the same pattern again, but two types are large and small trees, and this then is called the univariate pattern. And of course, at the end, you can have in a tropical forest, for example, hundreds of species, and this then is called a multivariate pattern. Yeah, so in point pattern analysis, usually one has four very important issues. The first one is to determine what is the most appropriate data type for the question and most of the data on hand. It seems to be trivial, but in some cases it's really decisive to have the right data type because all other steps depend on the data type. Some nodes might be completely different for different data types. Yeah, once one knows the data type, one can select appropriate summary statistics, and they are needed to quantify the statistical properties of the spatial patterns. But of course, we want to go one step further than simply quantifying the spatial structure. We want to respond to some deeper questions. And for this, we need null models. The null models, these are point process models with no structure, and they can have different purposes. For example, they can be in the sense of a classical null model, they can be the randomization of certain aspects of the data simply for detecting a pattern in the data. This could be, for example, to find out whether the univariate pattern is completely random, or if there's some clustering in the pattern, or some regularity in the points. But often this is only the first very simple step. For more complex patterns, one needs more refined methods, and these are provided by point process models, parametric point process models that can be fitted to the data, and then the parameters of these models contain some information, condensed information on the properties. And we will see very soon one example for this case. Yeah, then also the null models, or the point process models, they can be a representation of the ecological mechanism or the ecological hypothesis to be tested. And then it's clear, if you have summary statistics, if you have a null model, and this null model is usually one that must be simulated, then you need also some techniques to compare the data with the simulation of the null model. So these are basically the four steps one has to do in this type of analysis. Yeah, I like to illustrate you some of the cutting edge work in this area with my current ESC project. So I thought it might be better to give you some idea about my current work and not a type of review of all different things that have been done in this field. Yeah, and just a few words to this type of ESC advanced grant. This is a quite competitive thing. There are only very few ecologists that have the honor to receive this thing of grant. And it's a new, relatively new funding scheme by the European Research Council. And in contrast to the other European community network projects, it's focused on individual principal investigators. And it funds the research team for ideas, scientific ideas of the principal investigator. And there's also some money involved with this. It goes up to 2.5 million euro in 5 years. Yeah, some brief introduction to this project. You can see how the spatial point pattern analysis fits into the bigger picture. The big question in ecology is persistence of species, especially in species-rich communities. And you all know, 10 years ago, there was a big scandal in ecology. This was provoked by a book published by Stephen Howell on the unified neutral theory of biodiversity and biological traffic. This was in some way quite a shocking book, because here Howell claims that neutral models, where all species are functionally identical, can explain important properties of species-rich communities. Yeah, and this theory was in a way very shocking because it contradicts most of the things that ecologists learn about species diversity and maintenance in communities. And also it proposes very strong simplifications on all the different processes that shape ecological communities. Yeah, some examples for this. These are data from huge tropical forest plots, and they show on the x-axis, they show the abundance of the species, and the y-axis, they show the number of species that have this particular abundance. And the species abundance distribution is one of the key summary statistics in this type of work. And as you can see, the fit with the neutral model is really good. And this is something actually challenging to understand why does this very simple model provide such a good fit. And one interesting thing is that most people before Hubble, they looked at differences between species. So they said, this is a canopy species, this is an understory species, this is a light bending one, this is a shade tolerant species. So they focused on differences. But Hubble said, I want to take an opposed point of view. I focus on the similarities between species. And then I ask, how far can I go to explain important structural properties of ecological communities by looking only at the similarity of species? Yeah, so this is the question, which processes govern composition and dynamics of species-rich communities. And our contribution or our idea to this question, with my project, is to adopt a novel, spatially explicit perspective. So as I said before, many processes and mechanisms have a very strong spatial component. So then, why not using all the information which is buried in this enormous spatial data sets which are not available? So the first step, the first part, the key parts of the project is to quantify the spatial information which is buried in this data sets. And then the second step is that we also develop dynamic forest simulation models, ranging from very simple to very complex models. And all these models have outputs which are equal to the structure of census data. And then we can compare model outputs and census data and can ask which mechanisms, structures, and processes are really required to match all the multiple observed spatial patterns in real communities. Yeah, so basically the project has these three parts. And I focus here on the first part and give some idea on the last part. Yeah, so there are data sets available. Here you see a map, mostly in tropical forests, but also in temperate forests. Each of the dots here represents one huge 25 to 50 hectare plot of forest. And in this box, every tree from up the size from 1 centimeter DBH, so this is really nothing, is mapped, measured, and censored every 5 years. So each of these plots has more than 100,000 trees, and many, of course, many different species. So here's some example of a plot in Srinagar in Sri Lanka. On the left-hand side, you see all trees larger than 10 centimeters, so these are only the larger trees. On the right-hand side, you see some idea what species, the different species are distributed in this forest. Yeah, some really big data sets, and then you need to quantify these complex spatial structures. And this is now my first example. These are univariate analysis. This is only one type of point. And this question is how to quantify these complex spatial patterns. And in tropical forests, it's well known that many tree species have a clustered spatial distribution, and also that many different processes may cause clustering. So dispersal limitation, clump seed dispersal, forest gaps, or many others. And of course, one cannot expect that all of these processes have their critical scale at the same magnitude. So it will be quite logical that one may observe clustering at multiple scales. So these patterns may have different critical scales of clustering. And then the fundamental question is, are the properties of these patterns, of these cluster structures, are they in some way related to species properties? And this is the thing here I want to stress in the first example. Yeah, for this reason I developed a point process that incorporates two different scales of clustering, and this is based on the work here in 2007 in ecology, and then extended in one in 2009 in American Nationalist. And I use the Thomas process as a basic process, and this is the simplest possibility to represent the cluster pattern. And this works very simple. And the principle is that points are replaced by clusters. And then this is repeated in several generations, and then you have a clustered process. So one starts with a random pattern. So it's a random pattern, and then each of these clusters, of these points, is replaced by a cluster of a determined average size. And then inside this circles here, we distribute the points of the pattern. So these are now cluster pattern. And this is a classical Thomas process, which has one critical scale of clustering, which is indicated here by this small circles. And then in the next step, these points become a cluster again. If here you see this second scale of cluster, has the smaller circles. Yeah, there are two simple rules that specify this process further. This is that the number of points per cluster is a random, this is a Postpoint distribution, so basically points are randomly assigned to clusters. And then the distance between a point and the center of the cluster of two species placed relative to each other. And this is a quite important thing, because this determines the inter- and intraspecific interactions. So if the species don't meet, like this case here, where you have two species which are completely segregated, even if they are relatively close to each other, they segregate, then they have no chance to meet, they have no chance to develop some interactions. But one also may have some species which are distributed more or less in the same areas of the plot, and also if they are close, they are even a bit attracted. And these structures are the ones of interest here. And then, of course, one has to repeat this for all pairs in a community. And this, the whole sum of them then gives the co-distribution patterns. Yeah, and then the question is, does this co-distribution pattern change perhaps with life stage? We may expect for recruits, could be like the Pacala segregation hypothesis. We may think that recruits of different species may be placed in segregated clusters, so it may be segregated. And then we may expect, for example, that the pattern becomes more and more neutral when we approach the adult stage. Yeah, and then finally, also an interesting thing is whether species properties influence the co-distribution pattern. Yeah, and to resolve this question, I use two of the fundamental summary statistics in point pattern analysis. One is related to Ripley's K function. This is basically the mean number of trees of a second species in the neighborhood of individuals of a first species. So this gives the average association, so whether species 2 is more or less frequent than expected by chance in the neighborhoods of the first species. But there may be cases where this mean can be quite unequally distributed. For example, we may have a case where some individuals of species 1 have many neighbors of species 2, and others have no neighbor at all. In total, the mean may be still positive, but it's unequally distributed. So to distinguish these cases, we need a second summary statistic, and that is the nearest neighbor distribution function. So the probability that trees of species 1 has no neighbor of species 2 within this certain distance. Yeah, and these are the equations for the two axes, and then I make a two-dimensional scheme to classify the association of species pairs. Yeah, and then I have basically 4 different possibilities to combine this
these two summary statistics. And additionally, of course, I have one case without spatial patterning. This is the case where the second species is basically randomly distributed with respect to the first species. So this is the no association type. And then I have this mixing. These are when the two species are much closer together and consistently closer together. Segregation, where the two species are consistently segregated or repulsed. And then we have partial overlap. This is the situation where in some parts they are mixed, in other parts of the plot they are segregated. Yeah, and there's also fourth type, but this almost never occurs. Yeah, and I test these questions here with data from the BCI plot. This is a quite diverse thing, and this is, say, the father of all these big forest plots. It was funded more or less 30 years ago. This was a quite strange idea at this time to map a 50-hectare plot where all people mapped only 1 hectare. But at the end, it was a quite successful thing. And I look at 3 different size classes. First, the recruits, then juveniles. These are all individuals which are not recruits, but which are smaller than a certain reproductive size threshold, and adults are all the others which are larger and not recruits. Yeah, so how does the co-distribution pattern of the whole community, how are these 4 or 5 different types are distributed with scale? How does this change? So this is the pattern of recruits. So you see in the x-axis, you see the neighborhood, the radius of the neighborhood. And at the y-axis, you see the proportion of all species pairs. So there are several thousand of individual analyses. And you see the proportion of species pairs that belong to one of these 5 different type of association patterns. And there was some big surprise, which we actually did not expect. We found that about half of all recruit pairs show mixing, and the other half does not show any association. So there was no sign of segregation at all at small scales. And this was a really, I would say, a bigger surprise. Yeah, and then if the scale is something larger, then the mixing disappears, but on the other hand, the partial overlap becomes more important, and also the segregation increases on cost of the no significant association type. So this is the pattern for recruits, and now we look how it looks for juveniles. And there we see 2 tendencies. The no association type increases quite a bit, and the mixing type decreases. And also the segregation thing increases. So it looks there are really fundamental shifts in the spatial organization of this forest with different size classes. And then in the end, reproductive individuals, they are dominated by the no significant association type. So this is up to 80% at small scales. The mixing almost disappears. A bit of 9% or something like this, of course, remains. And also the segregation becomes a bit more important. But still, the dominant thing is the no association type. Yeah, and then in the next step, I want to look a bit more in detail on species properties. And I first show you the intensity of all recruits in the plot. So this is here a fit with a moving window of, I think, 50 meters. And this shows you that the recruits are heavily clustered. And all these clusters are probably canopy gaps in the forest. And now this is the pattern of all recruits. And now we look how these patterns change if we look only at canopy trees, mid-story and understory trees. So these are now the canopy species. And you see this pattern is much more clustered than the one of all recruits. And mixing, the positive association is much, much stronger. It goes up to 70% at the smaller scales. Then if I go to the mid-story, the strong mixing disappears, the no association type increases, and then finally the same things continue for the understory. So the understory is much more diffuse, although still has strong clusters. The no associative type is quite strong. So it becomes obvious that some of the species properties, of course, play important role in the spatial structure of recruits. But it's in some way funny that understory, midstory, canopy trees, this is something that characterizes an adult large tree, but this already has some influence on the recruits, on the very small trees. Yeah, so the main results quickly. The recruits community is dominated by positive co-distribution patterns. The adults show a fundamentally different co-distribution structure. It's dominated by neutral patterns, and species traits left a strong structure on these patterns for recruits. Yeah, so in some way it looks that this non-neutral pattern for recruits, but then at the end it ends up in approximately neutral pattern for the adults. So this is in some way, I would say, a surprise. Yeah, what are possible underlying processes? Of course, with this type of analysis, it's not possible to identify the underlying processes without any doubt, but one can put forward some strong hypotheses. So we certainly can exclude some processes that lead segregated patterns, like the segregation hypothesis of Pacala or the regeneration niche theory. But the patterns agree in some way with expectations of something that is called herd protection hypothesis. This is something similar to the Janssen-Connell hypothesis. And this says that monospecific clumps of small seedlings have a disadvantage because of shared pests and enemies. So they have a higher mortality in this case. And if one has an individual in a heteroid-specific clump, this has a slight advantage in survival, and this can at the end result in this multi-species clump pattern. But of course, one needs some additional mechanism that causes the initial clustering of the patterns. And one hypothesis would be the aggregated seed dispersal. This is a very good candidate because many of the tree species at PCI, they are animal dispersed, and many of these animals, they have places where they dump many seeds of many species together. Or of course the canopy gaps, they also may be an important structure that can promote multi-species clumps of recruits. Yeah, so this was my second detailed example, and the third example is only a very quick thing, to give you an idea how one can quantify spatial structures in diversity. So this is, again, all large trees in the Sinaraja forest, and now there are hidden structures in diversity. Now I show you the local diversity in 10-meter neighborhoods. And you see there are quite marked distributions of diversity. There are some areas that perhaps have only 5 different species in these neighborhoods. Others have up to 25 species in this neighborhood. So, which factors may cause this very strong variability? So we may ask, are there some key species in this forest that act like an accumulator of diversity, that have very positive interactions to other species and attract other species? Or are there some diversity repellers which basically avoid or make avoidance of other species in the neighborhood? Or is this an effect basically of habitat association, that some habitats are more diverse than others? Or is this related to tree density, that some areas have a very high density of trees, which then has a very high density of individuals and species? Or is there some locally dominant species? Yeah, and to address this question, I created my own summary statistic. This is called the individual species area relationship. This was presented in a 2007 paper. And just briefly give you an idea how this is related to all the different summary statistics you know. So the Ripley's K function. So I always ask how this person Ripley managed that the summary statistics was named after him. So I don't know how he did this. I guess he did it not himself, other people did it. So then I sincerely hope that you, if you should at some point use the individual species area relationship, that you may call it Wiegand's individual species area relationship. Well, so Ripley's K function basically describes the number of trees of a second species within a certain distance of a typical tree of the first species. Then you know the typical species area relationship. This is the expected number of species in a radius, certain radius, in a certain disk. So this species area relationship is not point or individual centered, it's location centered. Ripley's K function is centered about a focal species, and my individual species area relationship now is the number of species within distance R of a typical tree of the focal species. So in some way it's a mixture between both of them. Yeah, and here one example, this is a species which is clearly located in highly diverse habitats. So here the individual species area relationship is, has a much higher value than expected by a null model where I distribute the species randomly over the plot. So up to 8 more species in a 50-meter radius. But if I account for these environmental differences and displace in the null model the focal points only a few meters, say 50 meters, then the whole positive effect disappears. So this case is clearly an effect of habitat preferences. And then I have another species. You see the species are more or less equally distributed in low and high diversity habitats, and this is one that is completely neutral in a way, so it has no positive or negative associations. Yeah, this is enough to this type of analysis of species, individual species area relationship. And finally, a brief idea how to integrate spatial pattern analysis and dynamic simulation models. Yeah, so we have forest simulation models which produce the same type of data as the census data, so we can make a one-to-one comparison with census data. And this is a representation of the models, and here you see some typical simulation of how these forest models work. And here the summary statistic is the basal area, but you can use any other, for example, many spatial statistics. And you see also there are quite strong spatial structures appearing in this plot. So we will use this type of model, this four-mind model for this. And we have actually already some things of this implemented. Yeah, and then we are confronted with the problem that we have to do statistical inference with simulation models. So you all know statistical inference with statistical models, like regression models, but now we have to extend this to simulation models. And there are 3 steps. The first step is we have to quantify the spatial patterns with summary statistics or other patterns. So these are the properties of the data. Then we have to measure the distance between the observed data and the simulated data with some measure, and this is here done with the likelihood function. And then in the final step, we need some intelligent methods to find minima in the parameter space and to do the model selection thing. And then one can use this with MC and some Bayesian techniques or approximate Bayesian computation things. Yeah, and here we are already on the way. We have a recent review in ecology letters where we present all these different techniques for this. And we also have some example where we use these techniques, actually also in a forest model, only one species, but this worked quite well. Yeah, so this is already the end of my talk, and I see I'm quite well in time. So I had these 4 examples of my project, and I hope I give you a few take-home messages. And the first is that spatial point pattern analysis is much more than the usual K-function complete spatial randomness package that most people use. So you can do much more things. And these techniques provide very sophisticated methods to reveal spatial structures, can help to derive very specific hypotheses on the underlying processes. And also, one is now at the way to make an integration with dynamic simulation models to actually really use the full potential of these methods. Yeah, thank you very much for your attention.